Once you've done an analysis like a multiple sequence alignment, and you can see that there are differences between two groups, and in this case, it would be those proteins that are from isolates that are susceptible or resistant to a particular antibiotic. How do you know if those results are significant statistically that you could use in a publication? Well, BV-BRC has a tool that you can use that will show you that, and it's called MetaCats, which stands for Metadata-Driven Comparative Analysis Tool. And that's what we're going to talk about right now. So it's looking at the statistical analysis of individual SNPs. So there's specific features of MetaCats in that it gives you this statistical comparative analysis, and it's using a chi-squared statistic. And it's also looking at p-values within those. It can use either nucleotide or amino acid data. And there are three ways you can input the data. You can give it a group and ask it to look for specific differences based on metadata. You can use feature groups that you've created in BV-BRC. And you can also bring in alignments. So these could be alignments that you've generated outside the resource or alignments that are produced by other services within the resource, like MSA, like GeneTree. So there's different ways you can reuse this data. The publication associated with this is from 2013. So if you want to read it, and it gives you more information about the tool. So the MetaCats method, it creates a multiple sequence alignment if you're not bringing in your own. And the two methods it uses are muscle and UCLUST. It's doing chi-squared tests of independence, and it's also doing a Pearson's chi-squared test. And the Pearson's is used to calculate the p-value to show you the level of significance. So what you get back are the positions and their statistical significance. It's not like an MSA going to show you all the positions. If they're all the same, if there are no significant differences, it's not going to show you that. It's only going to show you the ones that have statistical significant differences. And in that table, it'll show you the aligned position, the chi-squared value, the degrees of freedom, and the residue diversity between groups. I find this really valuable, so I can look at things more cleanly. So the steps on the chi-square, it's a test of independence at each non-conserved position. Remember, it's not going to show you the ones that are all shared or are so weakly insignificant. And so the null hypothesis that it's starting with is that these groups you've created, that the SNP positions have no relationship at all to those individual groups. And then the chi-square is trying to disprove that. If you submitted DNA sequence, it's assuming that at any position, it can be any of those nucleotides. So that would be the 4 of them. For the proteins, it will look at all the amino acids across the group that are at an individual position, and it will use those to determine the degrees of freedom. And when the null hypothesis is rejected, meaning that it's proven that the group that you created or the metadata that you're looking at, like maybe the isolation country or the year it was isolated, when it's rejected, it means that there is something significant about that. So then it will do this pairwise chi-squared calculation. This is how you submit the job, and it looks complicated, but it's really not. One thing you can do is specify what you want the p-value to be. The default is 0.05, but you could go to 0.01, 0.001, or higher. If you use auto grouping, it will look at these particular values. This tool was originally set up for viral sequences. So sometimes you'll see things like the H1, H3, and H5 clades are for viruses. But this tool is also very useful for bacterial proteins. Especially, I found it useful for the antimicrobial resistance proteins. And so to find those things, you click on the down arrow under metadata and you select whichever of those you want with the group that you're going to use. You can also use feature groups. This is the one I use a lot. I create different groups in the resource for the proteins and I compare them. And that's the main thrust of what we'll be doing in this series of videos, although I will show how to do each of these things. Now you can type in the name of the feature, or you can click on the down arrow and select it. It's going to show you the feature groups you've created from most recent to less. And then if you have an alignment file, both from within the resource or without, you can put that in, but you will have to specify the metadata that you want with this file. So I'll show you how to do that as well. So that generates job results, and we'll step through these. It gives you the chi-square table, and when you click on it, this is what you will see. It will return something like this, and you can see that it's got a bunch of values with the position. It shows you the chi-square value, the p-value, the degrees of freedom, and then the last columns are the groups you've looked at. This will be 2. You can do more groups than that at a time. It gives you a MC table, so the multiple comparison p-value, so that you can look specifically at the p-value associated with each of those positions. It gives you a math log. We've talked about this before. It's just stepping through what the tool did as it ran through the alignment series. And it gives you an output, which in this one is an AFA file that's showing the aligned proteins. So the example data we're going to use is based on this publication by Stanhope et al. From 2008, and they looked at the penicillin-binding proteins. We're going to specifically look at PBP2X, but for some background data, each bacterium has 6 penicillin-binding proteins. Originally, they were called that way archaically because of the action they have when penicillin is present and it's knocking out these specific proteins. But what they really are transpeptidases, and they're helping build the outer membrane of the bacterium. So they're essential proteins. And if the bacterium is missing some of these, it will not be able to survive. And those big brown blobs that you see there, those are those penicillin binding proteins. And it's taking 2 of the glycopeptide chains and joining them together to provide this cross-linking so that the outer membrane of the bacterium is created. When a penicillin type antibiotic comes in, it has a similar structure to what is happening when the 2 peptidoglycan chains are bound together. And it's able to go into that transpeptidase and stop it from proceeding on. So that's why for a bacterium, it's devastating. For a human that's infected with this bacterium, it could be a lifesaver. So the function of PBP2X in particular is this transpeptidase catalyzes the last steps in this peptidoglycan cross-linking during cell division, specifically at the septal cross wall. Each of the 6 have a different function, although in some cases, some can fill in for the others if one of them is damaged. If you delete PBP2X, it leads to significant cell morphology defects. The cells can't divide well, they get swollen, and the mother and daughter cell die. If you're the human that's infected, that's good news. If you're the bacteria, that's it for you. Now, you can alter the PBP2X and have it have lower affinity for beta-lactam antibiotics. You wouldn't want to do this from our perspective because that leads to resistance and sometimes devastating resistance. But if you're a bacterium, this is really important for you if you want to survive. So PBP2X is especially important to things like cephalosporins, which are a class of beta-lactam antibiotics. And there are specific mutations that we're going to look at today that occur at the active site to reduce antibiotic binding. So I like when CARD has information. It's really nice because it spells everything out in a nice, clear way. And they do have even a landing page for PBP2X and Streptococcus pneumoniae in resistance to amoxicillin. And look, they also include at the bottom that Stanhope publication. And they also give a list of the mutations, and they give you a sequence showing where those mutations are within it. So what we're going to do, what our task is, should we choose to accept it? And obviously you're watching these videos, so if you don't watch the next one, it means you didn't choose it. And if you do, you do. We're going to examine the differences within these proteins from isolates that are susceptible to amoxicillin and those that are resistant to amoxicillin. We're going to look specifically at some mutations that were identified in Stanhope et al. As being important in determining resistance. And then we're going to look at those on a protein structure in BV-BRC. This tool, at first, I wasn't a fan of it, but I love it now. I use it all the time. It takes me to places where I get stuck at a multiple sequence alignment or at a gene tree. It shows me actually how these things are changing and where they are. So I love it, and I hope that you will too. So join me in the next one, and we will submit the job. See you there. Bye.